All right, so in the next section, we'll take a look at proteins, amino acids, and enzymes. And the first one here, we're looking at classifying amino acids. And remember, for this, you will be provided with a list or a amino acid chart. You don't need to memorize the structures. This is asking you to look up and write down the name of a naturally occurring hydrophobic amino acid. All right, so remember, amino acids, AA, are classified based on their R groups. And the R groups are just the side chains that make the amino acids different from each other, right? So if you look at the R group, that tells you a lot about that particular amino acid. You can find it here in the data tab. Let's go down to see if we can find our amino acids. Where are they? Naturally occurring amino acids. All right, so the R group for these are typically shown poking down here at the bottom in this particular chart. So there's the R group for alanine, there's the R group for valine, there's the R group for leucine, for isoleucine, phenylalanine, etc. And these are classified based on their polarity: non-polar, polar neutral, acidic, basic. So hydrophobic is just another word for non-polar. So if you look, for example, at the first category, non-polar amino acids, any of these would be sufficient. So if you take a look at alanine, for example, the R group here is a CH3, right? And a CH3 itself is just made up of carbons and H's, very non-polar, or things that have very similar electronegativities. So the R group is going to be non-polar or hydrophobic. So that would be an option. Any of these here could be options for hydrophobic amino acids, right? So glycine, for example, would be an example. All right, so in this one, it's asking us to look at peptides, describing them using their three-letter codes. So for example, it says answer the questions about this molecule. So you're given the structure, which I've copied over here. And it's asking what kind of molecule is this, right? There are different things we can look for in the molecule that will allow us to categorize it. What you'll notice is that there is an NH3, there's an amino end, there's a carboxylic acid end here on the right side. And the other things that we can note is that there are amide bonds. There's a C double bond ON, C double bond ON, C double bond ON. The amide bond is the same as a peptide bond. Right, that's just another name for the same type of functional group. So we have 3 of these. So this would be an example of a peptide. So if you said it was a peptide, write a description of it using the 3-letter code separated by dashes. So we need to identify the amino acids that make up this peptide. Okay, amino acids are identified by their R groups, right? So we need to find the R groups in our peptide, and by identifying the amide bond, we can quickly see the alpha carbon, right? We'll circle the alpha carbons in yellow, because that's where the R group is going to be connected. Okay, so our R group here for this first one is just going to be a simple H, right? And that's one of the H's that we have in the CH2 here. Same thing for the second one. We have an H there as the R group. For the third one, we have this thing, CH2 connected to a benzene ring. For the fourth amino acid, we have all of this as our R group. So what we need to do, right, is to look up the R groups from the chart, from the amino acid chart, to identify them and specifically to find the 3-letter code that corresponds to these. So if we look these up, what we'll find is that the amino acid with H as an R group, that's glycine, which is GLY. This is also glycine right after it. This here is called phenylalanine, which is a PHE. And then the last one here is called asparagine, which is AS. in, right? So you'd put those in, three-letter code separated by dashes. All right, so now we're going to try to identify some of the interactions between the residues in a protein. And when it comes to this, what we're really looking at is the interactions between the R groups, right? So we're looking at R group interactions. In order to do this, you would actually need to look up the structures in your amino acid chart of the amino acids in question. And R1 and R2 are the amino acids that we're going to be comparing. We're going to look specifically at their R groups to see whether there's any possible R group interaction between them. Okay, so we can do this, the third one here. So between serine and another serine, let's look at the structures of both of, well, serines to begin with. And we can see that serine, which is this one here, has an OH as part of its R group. So that's a hydroxyl group. We have another serine with a OH as part of its R group. They can interact with each other, right? And the interaction between them, when you have OHs, right, they can form hydrogen bonds between each other. So if you look here, right, we'll see, yes, there's an R group between them, a hydrogen bond. If we look at the lysine and glutamate, for instance, right? So if you look up lysine, so lysine is down here. So lysine has the NH3 plus, there's a positive charge on this R group. Glutamate has a negative charge. The interaction between them, positive and negative, is nothing more than a salt bridge, which again is nothing more than an ionic bond, right? But then we call that a salt bridge between those two. So you would need to look up the structures of the amino acids, specifically their R groups, and then try to see how they can interact with each other and name the type of interaction between them. All right, so in this question, we're taking a look at the different levels of protein structure. And as a general theme in chemistry and in biology, there's a pretty significant connection between structure and function. That is, a molecule or a compound needs to have a very specific structure in order for it to carry out its function. And that's especially the case for proteins. And there are different levels of protein structure, as you will learn or have learned in this particular section. So we're given a statement. We need to decide what level of structure, protein structure, is being created, destroyed, etc. And we also need to use our little slider here to diagrammatically show those changes. So it says enzymes rapidly lose effectiveness as temperature rises and they begin to denature. So what is denaturing? Denaturing is the loss of enzyme activity due to the loss of the three-dimensional shape, right? So the three-dimensional shape is the tertiary structure, and that is being destroyed when an enzyme gets denatured. So there's the three-dimensional shape is the folding of the protein on itself, and then it is going to essentially just extend out to look something like this. So you need to move your slider to get to the correct indication or the correct diagram. Second one, after a new protein is built from amino acids, protein folding begins, and the final step, smaller structural elements like alpha helices, beta sheets take on the final arrangement. All right, so here again, because the final arrangement is for the protein to become functional, that's your tertiary structure. So that's being created. Now we're actually starting off with the alpha helices, beta sheet that look like these, and then those become into the folded compact shape of the protein. Last one, after a new protein is synthesized, the final step is to gain function for the protein to fold up into its native form. So again, the function is dictated by the tertiary three-dimensional shape that's being created, and that's going from the initial primary structure to the folded shape of the protein. All right, so in this question is we're taking a look at some of the basic facts about enzymes and how they work. And then we're given, in this case, 3 different scenarios. You can press the little play button to see what's occurring. in each of these. And the key thing about enzymes is that they are catalysts, all right? So they speed up reactions, but they themselves are unchanged from the beginning to the end. So the key thing here is that if you start off with an enzyme, at the end of the reaction, you still have the same enzyme, right? So it should be there at the end. So the question is, do any of these sketches represent the action of an enzyme? And we would need to play our videos to see whether we still at the end of the video have the same molecule that we started off with. All right, and there's only one of these that does, and that's number 2. This one here, we start off with this blue thing with the hole in it, and then we end up with the same blue thing with the hole in it. None of the others have that. So at least number 2 is going to be an enzyme. What does A represent in number 2? So A here is the thing that the enzyme is working on, and that's called a substrate. B is the enzyme itself, and C is the region in the enzyme where the action actually happens, the space where, or the region of the enzyme where the substrate actually binds, and that's called the active site, right? So this is just gauging your ability to identify an enzyme, and then also to locate different regions on the enzyme. All right, so the last one here looks at understanding some of the models of both competitive and non-competitive inhibition of enzymes. And this particular enzyme here has 2 binding sites, site A and site B, and it can exist in 2 different conformations. It can actually have 2 different shapes, state 1 and state 2, right? So you can toggle between these. This also says that the substrate, the normal substrate of the enzyme actually looks like this. Okay, so if you look at between state 1 and state 2, there's only one state in which there's the active site that actually resembles or that is complementary to the shape of the substrate, and that's state 2, specifically site B. All right, so in state 1, there isn't a place for the substrate to bind. In state 2, there is this site here. So the active site then would be site B in state 2. So it says now classify each of the following molecules. So based on the shape, which of these categories could it belong to? All right, so if you look at this shape here, in state 2, it's not complementary to any of these sites. What about in state 1? It actually does resemble, was complementary to site A. Now remember, the active site is B, right? So this molecule, whatever it is, if it does bind, it's going to bind to site A. It's not competing with the substrate for the active site, so it can act as a non-competitive inhibitor. This molecule here is not complementary to any of the sites, A or B, in any of the states, so we're not going to check anything for that. This third molecule kind of looks similar to, or it's complementary in shape to the active site in B, so it can actually compete with the substrate of the enzyme for this active site. So that would be an example of a competitive inhibitor, right? Because it resembles the shape of the substrate, so it'll compete for that active site. If you look at the last one, this shape here, is it complementary? Well, it's not complementary to any of the sites in state 2. How about state 1? Yes, it is. It's complementary to site A. Remember, the active site is B. Okay, so it's not going to compete with the substrate for B. It's actually going to bind to site A, and it's not going to compete, so it's a non-competitive inhibitor. It's not competing, so it's not a competitive inhibitor, and it's neither the substrate of the enzyme either. So as you go through these, right, you want to take a look and look at the shapes of the active site, the shapes of the substrate, and of the other molecules that are shown to see whether they can actually bind to either of the sites on the enzyme molecule.